Hello. Today we'll be covering immunological methods that you can use both in the clinic and in the lab. So an immunologist's toolbox is very important. It's important to know the techniques we'll be talking about today to be able to organize and outline your own experiments, as well as interpret the results of others. So knowing the methods that are used in research of the immune system and in the clinic is really critical. And many tools that immunologists use daily actually use immune-generated compounds like antibodies, which will be the main focus of what we'll be talking about today. In addition, it's really important to understand the animal models that are used in research, to understand the role of specific proteins or cells within the context of the whole organism. So today's learning objectives will be to be able to describe methods by which specific proteins, including antibodies, can be identified, to describe methods by which cell types can be identified and isolated by using antibody-based methods, to describe methods by which cell functionality can be assessed, both with and without the use of antibodies, and also to be able to describe methods by which animal models can be genetically manipulated to express more or less of a specific protein. Now let's briefly go through the outline of today's lecture. So the types of methods we'll be covering today will first cover acquiring antibody samples and producing what are called monoclonal antibodies. Second, we'll be talking about the applications of antibodies. Then we'll go into techniques to study immune cell responses that are not antibody-based experiments. And we'll close out today's talk by talking about various genetic techniques that we can use in model organisms to further study the immune system within the context of a whole organism. So first, we're going to be speaking about how we acquire antibody samples, and then we'll talk about how to produce monoclonal antibodies. But before we go into monoclonal antibodies, let's talk about, in general, how we acquire antibodies samples. So antibody generation is the assessment of antibody titers in patients or experimental animals. Now, studying antibody responses within a lab can answer a variety of different questions, and as well as studying antibody titers in patients can give clinicians a really good understanding of what is happening internally in their patients. So when we talk about the generation of a specific targeted humoral response against a substance, this is called immunization. So first, let's go how we isolate antibodies in patients or experimental animals. So we isolate antibodies from the blood. When we're isolating these preps of samples, this involves the analysis of relatively crude preps of what is called anti-serum. So serum is the liquid that remains after blood is clotted. So what happens is you can take a blood sample from a patient or from an experimental animal, and this sample can either allow it to clot, or you can add a substance that prevents clotting, and these two different techniques allow for the production of two different types of liquids. As I mentioned already, serum is the liquid phase that remains after blood is clotted, and plasma is liquid that remains when clotting is prevented. And what's important to remember is that antibodies are isolated specifically from serum and not from plasma. So this is a mechanism by which, again, you can get these samples from patients, you can get these samples from experimental animals. Now let's jump into immunization. So immunization is the deliberate induction of an immune response to obtain and test for immunological responses, such as antibodies. So an immunogen, which is any substance that elicits an immune response, is really critical in this immunization. So I just want to clarify some terminology. So an immunogen is a substance that elicits an immune response, whereas an antigen is any substrate that can bind to a specific antibody. So immunogens are also called immunogenic. That's a descriptor of immunogens. And all antigens have the potential to elicit an immune response and specific antibodies, but not all antigens are immunogenic. As previously discussed in other lectures, you might remember that route, dose, and form of immunization will affect the response itself. So if we jump back to this idea of immunogen versus antigen, it's important that an antigen on its own may not be able to stimulate an immune response against that specific antigen. So you need an immunogen that allows for the induction of an immune response, specifically a humoral immune response when you're looking at antibodies. So adjuvants are often added to immunizations. So adjuvants are substances that are added to immunizations to enhance the immunogenicity of a highly purified antigen that have insufficient immunostimulatory capabilities. So for example, if you wanted to immunize an animal to produce antibodies against a specific protein, and you were to give this protein to the animal and there was an insufficient immune response to this protein, you can add an adjuvant to stimulate the type of immune response that you would like. So if you reference figure A3 in your Janeway textbook, there is a great chart that summarizes different types of adjuvants, their composition, and the mechanism of their action. I don't want to go through the details of this chart because you can do that on your own time. However, I would like to note that each of the different types of adjuvants will elicit a different type of immune response based on the innate immune PRRs that will recognize them and the subsequent immune responses that are driven. So you can use your adjuvants to drive the humoral response that you would like to your specific antigen of choice. Now, if you think about immunization in general, this will generate a polyclonal response. Now, polyclonal antibodies, as their name suggests, is many clones targeted against a specific antigen. So this is a large mixed population of antibodies that bind to different epitopes on the antigen, and these sorts of antibodies are very cheap to produce. So you can imagine that during an immune response, as you've heard in other lectures, the humoral response generates antibodies that are specific to various epitopes on these antigens. However, when in the clinic for clinical applications or in the lab, it's important sometimes to use a different sort of antibody. And by that, I mean monoclonal antibodies. So this is a little bit different from what we've just been talking about, but it's based on a very similar concept. So monoclonal antibodies, as the name implies, is one clone of an antibody. So this is a single antibody species such that it binds to only one unique epitope on the antigen. And it's more expensive to produce, and you'll see why in a few slides once we go over the techniques to make immune responses, or to make monoclonal antibodies. So why is it important to make monoclonal antibodies? So their unique specificity is really the key of this whole concept. So there's a variety of applications that you can use for this, but in general, the main idea is that with a monoclonal antibody, you know the specific epitope that is targeted on this protein, and so you have a much better understanding of what this antibody will be binding to and what sort of response it will be eliciting. So you have a much greater control of the experiment and the variables. So various applications can be used with monoclonal antibodies. Clinically, there's a lot of therapeutic uses, such as in cancer, autoimmune diseases, inflammatory diseases, and a variety of other diseases. We will not be covering these therapeutic uses today. However, they will be discussed throughout various other lectures. Applications that we will be discussing today of monoclonal antibodies will include diagnostic tests, visualization tests, cellular identification techniques, cellular functionality tests, and techniques to deplete specific cell types within a model organism or within a patient. So now let's jump into how to generate monoclonal antibodies. So as you've heard when talking about the generation of a humoral immune response, each B cell or plasma cell has a very highly specific antibody. So, in order to generate monoclonal antibodies, we want to isolate one specific B cell of a specific specificity and then generate antibodies from this B cell. Now, one B cell on its own within a lab setting will not produce enough antibody for us to experiment with. It will also stop replicating after a while once it's outside of its host. In order to get over this barrier and in order to produce many, many antibodies from a single B cell, a technique has been developed where a hybridoma is made. So a hybridoma is the hybrid between a myeloma line and a B cell, and this generates antibodies against a specific antigen. The way that these are generated is you take spleen cells, so about 50% of your spleen is B cells in humans and in mice. And so you take spleen cells from a mouse that has been immunized with antigen A. So this mouse immunized with antigen A representing any antigen of your choosing. This will elicit a variety of clones of B cell clones that are generated that will target this antigen. This would be a polyclonal population. Then, once you isolate these cells from the mouse, you also obtain myeloma cells that lack antibody secretion and the enzyme HGPRT. So what's important about myeloma cells is that these are immortal cells. So they replicate continuously within a lab setting, and they won't stop proliferating. And it's important that they lack the ability to secrete any antibodies that they might have or antibodies that they might encode, because you want the antibodies that are made from this hybridoma to be specifically to derive from the B cells from your mouse. So you will take these two populations of cells and you'll mix and fuse these cells with an experimental technique we will not be going through today. And then you transfer these cells to what's called HAT medium. So within this system, immortal hybridomas will proliferate, and mortal spleen cells and undiffused HGPRT myeloma cells will die. Now, what this means is that you're creating a system in which the only cells that will proliferate and survive are those that have fused, the cells that you would like. So the spleen cells will die within the medium because they lack the immortal potential that fused cells gain from the myeloma lines. And the myeloma cells that have not fused with spleen cells will die because they lack the enzyme HGPRT. Which within the fused cells, they get this enzyme from the spleen cells to which they have fused. Then, once these cells grow out within the media, such that only the fused cells will survive, you will have a population of cells that are producing a polyclonal population of antibodies, where each different B cell hybridoma is creating a different type of antibody against a different antigen. Now, you can then put in your specific antigen A or the specific epitope, and then select for your specific clone hybridoma, and then you can use these lines to produce massive amounts of antibodies that you could subsequently purify and use in experiments. So, just to summarize, I want to again go back to this concept of polyclonal versus monoclonal, in that the majority of the techniques that we'll be talking about next require the use of monoclonal antibodies. So now let's shift over to discussing the applications of antibodies and a variety of clinical and laboratory techniques that rely on monoclonal antibodies. These will, as I said before, these will include diagnostic tests, visual tests, cellular identification techniques, cellular functionality tests, and a technique by which you can deplete specific cell types within a patient or animal model. Let's start with diagnostic tests, which are tests that assess the presence of a particular protein or the presence of a particular antibody reactive against a specific protein. So the first experimental technique that we'll be going through is called the Enzyme-Linked Immunosorbent Assay, also known as the Elisa. So this technique is used to detect a specific protein within a sample. So let's say you have a patient or an animal model and you want to see whether or not within the blood or a tissue sample from this patient or experimental animal, whether you have a specific protein that is present. So you can take your unknown sample. So here these two are described as sample 1 and sample 2. And you can attach them to a solid support system. So within this example, it will be the walls of a plastic multi-well plate. Then you can add monoclonal antibodies that are specific for the protein of interest for your experiment that are labeled either fluorescently or are labeled with a compound that can then be converted into fluorescence. And you allow them to sit there such that the antibodies will only bind to the proteins if the proteins are there. You can then wash away the unbound antibody. Then you can add an enzyme that makes colored product from added colorless substrate, such that within the samples that have the specific epitope present, these cells will create color, and you can read these out with absorbance. This assay is also really important if you want to compare the levels of different proteins within different samples. So for example, if one sample has twice as much of your specific protein of interest as another sample, you'll have twice as much fluorescence from the first sample. The next technique we'll be talking about is immunoblotting, which is used for identifying the presence of a given protein within a cell lysate. But it also avoids the problem of having to label large quantities of cells. So this technique is also called Western blotting. So to describe this technique, I'll be using an example of HIV. So if you wanted to see what kind of proteins were within your molecule or within your virus, you can take your virus, here as HIV, and you can disassociate it in SDS, which is a detergent. So it'll break apart all the bonds within your virion. And then you can take this dissociated virus, this is also called a lysate, and you can run it on a gel with electrophoresis. Now, with electrophoresis, all your proteins will be separated by size. You can see here that our green proteins, these small dots, are the smallest, and these yellow globular formations are our biggest. Subsequently, once you run these, you can transfer this gel onto nitrocellulose and overlay it with anti-serum. So anti-serum again, is the serum that contains antibodies. And then you can detect so if you were to put your anti-serum on this sample, the antibodies that are present within that solution will bind to the proteins that you've run on this gel. And then you can detect bound antibodies with enzyme-linked anti-IgG. So what enzyme-linked IgG means is that you will take an anti-IgG antibody that is linked to an enzyme that can then be developed and you can look for fluorescence. Not only can you use this technique to see whether or not HIV is within your experimental sample by using anti-serum specific to HIV, you can also use this technique to see whether a patient has anti-HIV antibodies by taking a pure sample of HIV, running it on a gel, overlaying the blot with your patient's anti-serum, and if you see antibodies bound, that would suggest that your patient has antibodies against this virus. Now, monoclonal antibodies are also can be used to test for blood typing. So humans have 4 different types of blood cells, red blood cells, called O, A, B, and AB, based on the different surface antigens that they have on red blood cells. For a variety of reasons, it's important to know your blood type. Medically, the ideal way to get a blood transfusion, if you need one, would be to get similar blood types. So in order to test to see what kind of blood type you have or your patient has, this technique is based on hemagglutination. So this is the clumping together of red blood cells that is caused by antibody binding to surface antigens. So you can add serum, you can add antibodies that are specific for your A and B proteins on the surface of your red blood cells. And if you were to add these antibodies to the mixture, these antibodies will bind to the surface antigens for which they are specific if they are present, and this will cause the clumping together of cells.